Before you begin, prepare the cell culture flask with the respective growth medium and equilibrate the flask in the CO2 incubator for approximately 30 minutes. While the medium is warming up, it is a good time to get the rest of your equipment ready. Pipettes and tips, cell culture flasks, disinfectant, a timer, the information paper, the water bath, and dry ice. Make sure you know the exact location of your vials before getting to the nitrogen tank. To pick up the cells, prepare a box full of dry ice. Standard ice will not be sufficient to prevent the thawing from already starting. Quickly place the vials in the dry ice. Put the rest of the vials back into the liquid nitrogen tank. To vent the vial slightly, open the cap about 1/4 of a turn, close it, and immediately place it back into the dry ice. When all the vials have been collected, close the liquid nitrogen tank and proceed with the thawing step. Before you begin, make sure that the water bath has a temperature of 37 degrees Celsius. From this point on, the timing is critical. The dry ice box containing the vial should be located just next to the water bath. Thaw only 1 vial at the time. Do not submerge the vial completely, but make sure the water level is reaching up to the height of the screw cap, as you can see here. Slowly move the vial in the water bath for about 2 minutes. At this point, remove it from the water bath and take a quick look to assess the status. If there is still ice in the vial, continue with the thawing. If the content of the vial is completely thawed, proceed with the starting culture. Place the vial on a tissue and cover it with a validated disinfectant using a squeeze bottle. Very lightly wipe the vial. Place the vial vertically in the center of the laminar flow hood and dispose of the paper used for disinfecting the vial. Pre-open the vial before picking up the pipette. Also, if not already done, pre-open the cap of the culture flask. Before picking up the cells, gently pipette the content of the vial up and down only for 1 or 2 times, not more. Transfer the cells in the flask gently. When the cells are in the flask, close it and distribute the cells evenly by gently moving the flask back and forth and from left to right. The flask is ready to be placed in the incubator for 16 to 24 hours until the medium should be replaced. You may now proceed with the exact same steps for the other vials that you may have to prepare. A good preparation, along with an appropriate speed and good technique, will make your cells happy. Learn more on PromoCell.com.